Hello and welcome everyone. As promised earlier in the part 1 of this video, I'm back with the solutions of CPET 2020 Zoology question paper. So in the last part of this video, that is in part 1, we solved question number 1 to question number 35. So in this video, we are going to continue the same series and we will solve from question number 36 to question number 70. As well as I will also provide a brief description about each and every solution. But one more thing students, ମୁଁ ଷ୍ଟାର୍ଟ କରେ ତାଠୁ ଆଗରୁ ମୁଁ ଗୋଟେ ଜିନିଷ କହିବା ପାଇଁ ଚାହୁଁଛି କି ମୋର ଜଣେ ଷ୍ଟୁଡେଣ୍ଟ ମୋତେ ମେସେଜ କରିଥିଲା ମ୍ୟାମ୍ ଆପଣ ଓଡ଼ିଆରେ କାହିଁ କହୁନାହାନ୍ତି କିମ୍ବା ହିନ୍ଦୀରେ କାହିଁକି ୟୁଜ୍ କରୁନାହାନ୍ତି ଇଂଲିଶଟା ଆମକୁ ଟିକେ ଡିଫିକଲ୍ଟ ହଉଛି। ମୁଁ କହିବି ଷ୍ଟୁଡେଣ୍ଟସ୍ ମୁଁ ଇକ୍ୱାଲି ଫ୍ଲୁଏଣ୍ଟ ଅଛି ଓଡ଼ିଆରେ ବି ହିନ୍ଦୀରେ ବି ବଟ୍ ଆଇ ଏମ୍ କିପିଂ ମାଇ ଭିଡିଓସ୍ ଏକ୍ସକ୍ଲୁସିଭଲି ଇନ୍ ଇଂଲିଶ। ଦିସ୍ ଇଜ୍ ଫର୍ ୟୁ ଷ୍ଟୁଡେଣ୍ଟସ୍। ତମେ ମାନେ ଆଗକୁ ପିଜି କରିବ ଏମଏସସି କରିବ ହୁଏତ ଓଡ଼ିଶାର କୌଣସି ୟୁନିଭର୍ସିଟିରେ କର କିମ୍ବା ଯାଇକରି ସେଣ୍ଟ୍ରାଲ ୟୁନିଭର୍ସିଟିଜରେ କର ଆଇଆଇଟିଜରେ କର ଯୋଉଠି ବି କର ସେଠି ତମମାନଙ୍କୁ ପ୍ରଫେସର୍ସ ମାନେ ଓଡ଼ିଆରେ କଥା ହେବେନି ଆସିକିରି କ୍ଲାସରେ ସେମାନେ ତମକୁ ଇଂଲିଶରେ ବୁଝେଇବେ। ଆଉ ପିଜି ଟାଇମରେ ଷ୍ଟୁଡେଣ୍ଟସ୍ ବହୁତ ବଲ୍କି ବଲ୍କି ବୁକ୍ସ ପଢିବା ପାଇଁ ପଡେ ଏପର୍ଯ୍ୟନ୍ତ ଯାଏଁ ସାୟଦ ତମେ ମାନେ ନୋଟ୍ସରେ ହଉ କିମ୍ବା ନର୍ମାଲ ଇଣ୍ଡିଆନ ଅଥର୍ସ ହଉ ସେମିତି କିଛିଟା ବୁକ୍ସରୁ ତମେ ମାନେ ପଢିଥିବ କିନ୍ତୁ ଏବେ ଆଗକୁ ପିଜି ଟାଇମରେ ତମମାନଙ୍କୁ ସ୍ପେଶାଲି ଫରେନ୍ ଅଥର୍ସ ପଢିବା ପାଇଁ ପଡିବ। ଫରେନ୍ ଅଥର୍ସରେ ଇଂଲିଶ ସାଙ୍ଗେ ତମେ ଯଦି ଟଚ୍ ରଖିବନି ଆକ୍ୱେଣ୍ଟେଡ ହବନି ତାହେଲେ ସେଇ ଜିନିଷଟା ତମମାନଙ୍କ ପାଇଁ ଡିଫିକଲ୍ଟି ହବ। ଏଇଟା ୟୁଟ୍ୟୁବ ହିଁ ତ ଅଛି ଯଦି ବୁଝିପାରୁନ ବ୍ୟାକ୍ କର ଶୁଣ ଆଉଥରେ କିନ୍ତୁ ବୁଝିବା ପାଇଁ ଚେଷ୍ଟା କର। ମୁଁ କହିବି ତମମାନେ ଇଂଲିଶରେ ହିଁ ଶୁଣ ସୋ ଦ୍ୟାଟ୍ ଏଇଟା ତମ ମାନଙ୍କର ହିଁ ଆବିଲିଟି ଇନକ୍ରିଜ କରିବ ଆଗକୁ ବଢିବା ପାଇଁ। ଆଣ୍ଡ ଇଂଲିଶ ଇଜ ଅ ମିୟର ଲାଙ୍ଗୁଏଜ। କେବେ ବି ଗୋଟେ ଭାଷାକୁ ନିଜ ବାଧା ହବା ପାଇଁ ଦିଅନି। So without any further delay, let's start the video. Question number 36. Assuming they all had the same number of carbon atoms, yes, which of the following has the maximum number of carbon hydrogen bonds? Okay, all the following options have the same number of carbon atoms. Okay, let's see what are the options. An unsaturated fat, second is a unsaturated fat, polysaccharide, and saturated fats. Everyone is having same number of carbon atoms, sorry. So what is the number of carbon hydrogen bonds found here? Okay, first let's see the case of unsaturated fat. Remember the difference between unsaturated and saturated. Always remember the difference. Saturated means saturated with hydrogen. I always tell my student to remember this, otherwise you will get confused. Saturated means saturated with what? Saturated with hydrogen. Saturated structures will have what? Will have single bond. Saturated structure will have single bond. Okay, unsaturated structures will have what? Double bond. Unsaturated, when there is a case of unsaturated structures, they will have what? They will have double bond. They will have double bond. Likewise, you can remember. So saturated means saturated with hydrogen, as I say. That means they will have single bond. Why saturated with hydrogen will have single bond? Let's see. Okay. Suppose this is a saturated structure and this is an unsaturated structure. So what will happen whenever there is a double bond like this here? So what will happen? The number of hydrogen will become less. Yes or no? Here are two hydrogen, but whenever there is a double bond like this, what will happen? Number of hydrogen will get reduced. So more number of hydrogen is where? In case of saturated. That is why I call saturated structures as saturated with hydrogen. You will never get confused which one is double bond, which one is single bond. Saturated means single bond and unsaturated means double bond. Okay, double bond. Now, what is given? It is given that every structure here is having what? Same number of carbon atoms. Every structure is here having what? Same number of carbon atoms. So an unsaturated fat. Okay, what is the structure of fat? Fat is a long aliphatic chain of carboxylic acids are called fats. Okay, long aliphatic chain of carboxylic acids are fat. Means this can be any chain like this. and it is having a carboxylic group. This is what we call fats. Okay, so here the number of carbon atoms are same in every structure. So which one will have more hydrogen? Let's say a structure of 6 carbons, for example, 6 carbon, for example. So 6 carbon here and unsaturated fat is here. Let's say here particularly 4 carbons are given. Okay, let me add 2 more carbons. H. Here also H. 3H more. Okay, among this saturated and unsaturated, which one is having more number of carbon hydrogen bond? Saturated will have. If we compare with polyunsaturated means more double bond will be there. Suppose I'm adding one more double bond here and one more double bond here. What will happen? This hydrogen, it will not be present. Okay, this hydrogen will also get removed, right? So unsaturated, obviously it will be no, a polysaccharide and a saturated fat. Okay, I'm considering a carbon skeleton of 6 carbons, right? A polysaccharide, let's say glucose. I have the structure of glucose in my slide. Let me show you. Yes, here is the structure of glucose. So we are also having what? We are also having a carbon oxygen bond here. Carbon OH bond is also present here. So obviously the straightforward answer for this will be option number D. That is, a saturated fat will have more number of carbon hydrogen bonds. Clear? Okay, now let's move to the next question. Which of the following cofactor is essential for the activity of acetyl coenzyme carboxylase? Okay, answer for this will be biotin. Option number B, that is biotin. Ab enzymes are, what are ab enzymes? Ab enzymes, remember, ab enzymes term is derived from ab. Ab is derived from what? Antibody. Antibody. And enzymes, this term enzymes is derived from what? Zymes is derived from enzymes. Enzymes. Okay. So antibody plus enzymes is what? Ab enzymes. So what are ab enzymes? Ab enzymes are those antibodies which can mediate some catalytic reactions. Those antibody which can act like enzymes are referred to as ab enzymes. So answer will be option number B. Antibodies that have catalytic activities. Next question. Which of the following statements is true about non-competitive inhibition? Remember students, whenever there is a context of non-competitive inhibition, always the value of Vmax decreases. There are 3 types of inhibition as you must have studied in your graduations, like there can be competitive, non-competitive and uncompetitive. Okay, there can be 3 types of inhibition. So, in case of non-competitive inhibition, remember what happens? The value of Vmax decreases. The value of Vmax decreases. So, answer will be option D. That is Vmax decreases. Here is a graph I have also kept here. Remember, the value of Vmax is lower. Okay, and Km remains constant. Km remains constant and Vmax value decreases. Next question. Which of the following compounds is a positive allosteric regulator of the enzyme pyruvate carboxylase? So answer for this will be acetyl coenzyme A. Acetyl coenzyme A, that is option number B, is a positive allosteric regulator of the enzyme pyruvate carboxylase. If you people don't know about the meaning of allosteric regulator, these are the regulators which bind to the enzyme other than the active site. We know all enzymes. Okay, all enzymes have a certain site in which substrate binds, right? Substrate bind. This site is known as active site. Active site. If there is some other site other than active site in which a regulator is binding, that site is known as what? Allosteric site. Okay, so moving to next question. P53 is associated with all of the following except, answer for this will be post translational modification. Remember, P53 protein What p53 protein is otherwise known as guardian of the genome. You must remember p53 is otherwise known as guardian of genome. It has a very important role in controlling cancer. Okay, it has a very important role in controlling cancer. Hence it is also the tumor suppression gene. It regulates programmed cell death. Okay, it regulates programmed cell death. What is programmed cell death? Every cell performs a fixed number of divisions. Like normal cell can divide up to 2 to 100 times or 80 times, like that, and after that they have to die. Everything on earth that is born, it has to die. But what happens in case of cancer cell students? The cells undergo uncontrolled division. Okay, they undergo uncontrolled division and there is no programmed death in case of them. Okay, and this uncontrolled division is due to the absence or due to the defect, I must say, due to the defect in this particular p53 protein. Okay, so p53 is associated with tumor suppression. It suppresses tumor in case of cancers. It helps in programmed cell death. If p53 protein is active in the body, it will help in programmed cell death. And how it is regulating the programmed cell death? By activating some sort of transcriptions. Means like there are other proteins in the cells as well, right? Which have to undergo, which helps in DNA repair, which helps in death, okay, which helps in autophagy. There are numerous functions in the cells. So these functions are possible only because p53 protein allows them to do so. So this p53 protein is also responsible for transcription. Your question is p53 is associated with all of the following, right? Three of the following, it is associated, except what? Except post transcriptional modification. It is not associated with post transcriptional modification. Clear? So moving to next question. When heterozygous black pigs are intercrossed, then what is the chance of the first two offsprings being black? It is given in the question that heterozygous black pigs. Okay, the pigs are heterozygous and black. Means what? We are getting a hint that in heterozygous condition, which trait it is expressed? Black color. So black color, the trait which is expressed in the heterozygous condition is what? It is dominant. So black color is dominant over here. It is given in the question. And black heterozygous pigs are crossed. Let's denote this by capital B. Okay, let and small b. Heterozygous. They are intercrossed. Okay, they are intercrossed. Then what is the chance of the first two offspring being black? We can perform a crossover here. Like two heterozygous pigs are here. Two heterozygous pigs we are crossing. And what sort of progenies we can obtain from here? One can be capital B, other can be capital B, small b, other can be again, sorry, capital B, small b, and the last one here is small b, small b. In F1 generation, what we are getting? We are getting three black pigs. Three black pigs out of four. Okay, so 3 out of 4 are what? Black. 3 out of 4 are black. What we concluded here, that out of 4 pigs, okay, out of 4 pigs, 3 will be black. 3 out of 4 will be black. What the question is asking, what is the chance of the first two offspring? It is asked about two offsprings. Means what? We will simply multiply. Remember student, whenever in the probability question, it is you are getting the word and. Okay, you are getting the word and, always multiply. Okay, here it is what given? Two offsprings. Matlab first offspring and second offspring. So what will we do? 3 into 4 into 3 into 4. That is equal to 9 by 16. Out of 16 offspring, 9 will be black. So out of 100, how many offsprings will be black? Now we can find out. If out of 16, 9 are black. So out of 100, how much offsprings will be black? Simply we can cross multiply and we can find out the answer. Means 9 into 100 by 16. That much offspring will be black, which will be nearly equal to 56.25. So option number B will be the right answer. Next, question number 43. Toll-like receptors, TLRs, play an important role in immune defense by recognizing option number A, that is microbial components. Next question. The macrophage rich mass found at the site of injection of an adjuvant is called. Okay, what is adjuvant? Adjuvant is something that is added along with the vaccine to increase the immunogenicity property. Along with vaccine, something is added, a substance is added, substance is added. That substance is known as adjuvant. Why adjuvant is added? Adjuvant is added to increase what? The immunogenic reaction, to increase the immunogenic reactions in the body. The question is asking the macrophage rich mass found at the site of injection of an adjuvant is called, answer will be granuloma. So what is granuloma? Granuloma is a type of clusters of white blood cells, that is macrophages. Okay, granuloma develops as a reaction to inflammation, infections or foreign bodies. Next question, question number 45. The major variability of MHC is found is, answer will be alpha chain. Okay, if we closely look at the structure of MHC, this is the structure of MHC. MHC, major histocompatibility complex. You must have read about this in immunology unit, right? These are the two domains. This is the alpha chain. Here it is the beta chain. Okay, this was class 1 MHC, class 2 MHC. Always remember the difference between class 1 and class 2 MHCs. This is frequently asked question and this is the most favorite questions of the examiner. So, major variability region is found where in the alpha chain. It has two portions, like this is the variable region and second one, this is the constant region. Here we can find the N-terminal region, here we can find the C-terminal region. The major variable region is found where in the alpha chain. Next question. The predominant antibody in saliva is immunoglobulin A. Remember, the saliva and tears, our tears, are rich in what? In immunoglobulin A. Next question, cytokines are not. Question is asking, cytokines are not what? Antigen specific. Here I have given some properties of cytokines. If you wish, you can take a screenshot and remember it as well. So what are cytokines? Cytokines are substances that released by the cell due to some infections. Okay, due to some infections or due to any stimulus. Okay, these are some signal molecules which are released by the cells. These are known as cytokines. Next question. Type 1 hypersensitivity is known as anaphylaxis. Question number 49. In myasthenia gravis, blocking antibodies bind to acetylcholine receptor. What happens in myasthenia gravis? I am telling you, students. Our muscle functions, whatever the movement is there, if we want to lift our hand, move our eye, blink our eyelids, everything happens because our brain gives some signals, right? Our brain gives some signals to our muscles to function. But what happened in case of myasthenia gravis? Our brain is giving signal, but the problem is in case of the neuromuscular junction, means the junction, the point at which the neurons and the muscles is meeting. Okay, at that junction is known as was synapse. And in these synapse, what happened? Here are acetylcholine receptors. The brain is sending signals and these signals are converted into acetylcholine molecules like this. The acetylcholine molecules are received by the muscles. Received by the muscles. Muscles by what? By the acetylcholine receptor. What happens in case of myasthenia gravis? Some specific antibodies does what? It binds to these receptors. It binds to these receptors, blocking the acetylcholine transmission. And that is why signal is not received by the muscular organ. Commonly it is seen in case of eyelids. Okay, commonly it is seen in case of eyelids. People who are suffering from myasthenia gravis have difficulty in blinking also. So this is about myasthenia gravis. Now moving on to next question number 50. Which of the following is an epigenetic factor of gene expression in eukaryotes? Remember students, what is epigenetics? Epigenetics is the study of inheritance of gene expression, study of inheritance of gene expression that is not involving change in DNA sequence. That is not involving change in DNA sequence but what? It is involving the effect of environment. Okay, so sometimes what happens, we know all the information of our body, body, how the body is functioning, everything it is stored in where? It is stored in DNA. And if we are changing any DNA sequence, if we change the sequence, what will happen? Protein will change, obviously. Protein change, and what will happen? Function will be different. DNA function will be different. What happens in case of epigenetics? The DNA sequence is not changed. The DNA sequence is not changed, but there is alteration in the protein. How this happens? Let's see. Suppose in our DNA, what happens? There are also non-sequential part, like the histone part is there. Okay, so what happens? DNA methylation. Methylation particularly occurs in this part. That is why what happens? There is change in the type of protein that is produced by the particular DNA. This can happen due to environmental changes. So answer for this will be option number B, that is DNA methylation. Question number 51. Which of the following is a post-zygotic isolation mechanism in speciation? Post-zygotic means after the zygote is formed. After the zygote is formed, behavioral change. No, behavioral change is something that is pre-zygotic. After, if two individuals who are reproducing, if they are having some change in behavior, if they are having some change in behavior, zygote will be formed. No, so behavioral isolation is pre-zygotic. Seasonal isolation, if breeding seasons are different for two species, they will not be able to breed and they will not be able to produce zygote. So seasonal isolation is also not possible. Fertilization failure. Yes, fertilization failure means here also zygote is not formed. So this is not answer. Hybrid sterility. Yes, this can be the answer. Hybrid sterility means if two individuals are reproducing, a zygote is formed, and the hybrid which is formed out of this zygote, it is not able to reproduce further. This hybrid which is formed because of this fertilization event, it is not able to not reproduce. Okay, so post-zygotic isolating mechanism, the answer for this would be hybrid sterility. Question number 52. The phenomenon of genetic drift is most likely to occur in a population that is, always remember, genetic drift always happens in a small population. So option number A. The first vertebrates appeared in which of the following periods of Paleozoic era? Answer for this will be Ordovician period. Age of fishes was our Silurian period. Okay, so before Silurian period, in the late Ordovician period, some of the vertebrates were observed. And this is Devonian. This is a printing mistake. This should be Devonian. Okay, this should be Devonian period. Anyways, next question. Chaperone helps in protein folding and assembly, yes. Protein stability, yes. Both A and B, of course. Answer will be C, that is both A and B. Deviation from Hardy-Weinberg assumption of infinitely large population size results in, remember students, whenever there is deviation from Hardy-Weinberg equilibrium, there can be a chance of genetic drift. There can be a chance of genetic drift. Okay, but in this particular example, in particular questions, what is given? It is given in a large population. It is given in a large population. And just now I said genetic drift always occurs in what? In small populations. So answer for this will be heterozygote advantage. Answer for this will be heterozygote advantage. In a population that is in equilibrium, the proportion of individuals showing dominant trait at a given locus having two alleles is 84%. The frequency of the recessive allele in the population is, all know from Hardy-Weinberg equation, that is P plus Q is equal to 1, where P is the frequency of one allele, Q is the frequency of another allele. Let the frequency of this dominant trait that is given, let it be P. What is given in the question? It is 84%. 84% means in a population of 100, in a population of let's say 100 individuals, 84 individuals are having this dominant allele. Means 84 out of 100 are having dominant allele. That is 0.84. We can say it. So what P is equal to now? It is given 0.84. We need to find what the frequency of recessive allele in the population. Very simple. Just replace 0.84 plus Q is equal to 1. So the value of Q will be 0.16. Okay, this is my answer, students, but I have seen many people differing from this answer. So if anyone is having any objection in particularly this 56 number of question, kindly give me the information in the comment section. It would be highly appreciated. Next, question number 57. The folding of sheets of cells, the migration of cells, and cell death are mechanisms of. Since in this question, three of the events are given. Folding of sheets is given, migration of cells is given, also cell deaths is given. These all are mechanisms of morphogenesis. Next question. The process by which developing cells achieve functional maturity and identity as liver or muscle or nerve cell is called, answer will be differentiation. Differentiation is what? Differentiation is the process by which the developing cells attain their functional maturity. We all were once upon a time a single cell zygote. But from this single cell zygote only, numerous types of cells are derived. Like our muscle cells is there, fat cells is there, bone cells. Every cell of our body is derived from this single cell zygote. And this is only possible because of the process of differentiation. Since the cells multiplied and different types of cell differentiated into different function, that is why we have such diversity of cells in our body. Next, question number 59. Which of the following cell organelles actively participates in animal apoptosis? Answer for this will be option number D, that is mitochondria. Although nucleus is also a right answer, but mitochondria will be the more appropriate answer. Yes, one more thing. What is apoptosis? Apoptosis is the same process, what I call programmed cell death. Programmed cell death. I just now explained programmed cell death in some previous questions, right? Programmed cell death is the process which is called apoptosis. Next, question number 60. A species facing an extremely high risk of extinction in the immediate future is called endangered species. Next, which of the following zones in the lake suffers from lack of light and therefore limited photosynthesis is available to take place? So this is the structure of the lake. Here we can see the cross section here. What are the different zones? Littoral zone is there, profoundal, pelagic, euphotic zone. So answer for this will be profoundal zone. What is littoral zone? Littoral zone is the zone present near the edge of the lake. Lemnitic zone, it is the zone that is open zone. This open zone is known as lemnitic zone. Next, other zones are like euphotic and aphotic zones. Okay, euphotic zones means this zone up to which, photic means light, up to which the light is penetrating well, that is called that is euphotic zone or photic zone. Okay, answer is asking what? Lack from light. Profoundal zone. Profoundal zone, or it is otherwise known as aphotic zone. Profoundal zone or aphotic zone is the zone where photosynthesis is not actively taking place. Photosynthesis is limited at this place. So answer for this will be profoundal zone. Next question. In which year Wildlife Protection Act was implemented in India? The year was 1972. Before this implementation, our country had only 5 national parks, and today the number of national parks in our country is 106. Okay. Next question. The part of the brain that has been shown to function like a biological clock is suprachiasmatic nucleus. Next question. Which hormone is secreted by placenta? ACTH? No. Progesterone? Yes. ACTH is adrenocorticotrophic or hormone. Adrenocorticotrophic hormone. Okay, trophic hormone. So it is secreted from where? It is secreted from the pituitary. Progesterone is secreted from placenta. Okay, progesterone is also secreted by the corpus luteum of ovary. That is a yellow color mass. Progesterone is also known as pregnancy maintaining hormone. Pregnancy maintaining hormone. It is also known as pregnancy maintaining hormone. Why? The role of progesterone is to maintain the endometrium layer of the uterus. The uterus has three layers. Inner is endometrium, middle is myometrium, and the outer is epimetrium. Right? So the endometrium layer should be thick for supporting the growing fetus. So progesterone helps in maintaining the endometrium layer of the uterus, and therefore it is secreted by the placenta. So answer will be option number B. Next, question number 66. Induced breeding technique is used in. The term is induced breeding, means breeding is induced. This is used in case of culture fishery, marine fishery, capture fishery, or inland fishery. Answer will be C, that is capture fishery. Next, question number 67. Which of the following is an amino acid? Answer will be option number D, that is proline. Proline is having an amino group, therefore proline is an amino acid. Question number 68. Proteins may be separated according to size by. Answer for this question will be option number C, that is molecular exclusion chromatography. This is otherwise also known as size exclusion chromatography, or we can say it gel exclusion chromatography. These are the other names of molecular exclusion chromatography. Next is question number 69. All the enzymes of TCA cycle are located in the mitochondrial matrix except. Answer is option number C, that is succinate dehydrogenase. Question number 70, the last question. To be a cloning vector, a plasmid does not require origin of replication. No, origin of replication is a must have in a plasmid, from where replication process start. Next is antibiotic resistance marker. It is needed. It is needed for the purpose of screening. It is needed for the purpose of screening the recombinants. Next, restriction site. It is also needed. Next, last is to have a high copy number. No, this is wrong. This is the basic structure of plasmid. Plasmid is having what? Origin of replication. It is having antibiotic resistant genes like amoxicillin resistant. Next, selectable markers should be there. A promoter site should be there, and restriction sites should be there for the restriction enzymes to cut. And say the same restriction enzyme should be used to cut the desired DNA also. And both are incorporated to form a recombinant DNA. Both are incorporated together, both are mixed together to form a recombinant DNA. Recombinant DNA. And this recombinant DNA then transferred to the host cell. This is the whole thing about your recombinant DNA technology. If you all are liking this explanation parts, please do let me know in the comment section. So this is all for today. Thank you all.